Hello, my name is Trygestad, a master's student working with Dr. Addie Thompson at Michigan State University in East Lansing, Michigan. As some producers are aware, there is a newer disease called tar spot. MSU and many other universities have been working to provide information to growers on what they can do to prevent or slow down the disease from affecting their production. This video showcases the work Dr. Thompson and I have been working on to find genetic resistance to tar spot. To start, I will explain just a little bit what tar spot is. Tar spot is a disease caused by the fungi Maize. This fungi reproduces through spore release. The spores form in fruiting bodies that are held within a protective stroma. We see these stromata when identifying tar spot, as they look like tar speckled over the leaves. This video shows a plot that's been heavily infected with tar spot, causing rapid deterioration of the canopy, excessive dry down, and early desiccation of the plants. In some instances, the stromata have necrotic regions surrounding them called fish eyeing that can add to the deterioration. In cool and humid conditions, this disease can spread from a few plants to an entire field in just 3 weeks and can cause up to 50% yield loss. Initial infection of a new field occurs when spores are blown in via wind dispersal. Once present, the spores can overwinter on corn residue. This type of infection typically occurs in late July and early August. Tar spot is new to the U.S. as of 2015. Depicted here is the expansion of the disease. In the U.S., the disease first took hold in the Great Lakes region, but has spread to Iowa, Minnesota, and recently into Canada. Here at Michigan State, we are researching genetic resistance to tar spot. Our project goals are to find lines resistant to tar spot by screening 2 diverse maize populations, map that resistance in the maize genome, and cross the resistant varieties into more elite material to be used in commercial lines. We screened diverse maize varieties from 2 populations. The first group was the Wisconsin Diversity Panel. These publicly available varieties come from a range of universities and industry breeding programs that are often some of the grandparents of the lines on the market today. The second group we examined was the germplasm enhancement of maize, or GEM lines. These varieties each contain a mix of tropical and temperate maize. Tar spot is new to the U.S., but not to Latin America. In Mexico, it has been present for over 100 years, so some of this material may harbor resistance. We took ratings weekly using the scale shown, and we had fields in Michigan, Indiana, and Wisconsin. Now moving on to what we have found so far. The animation on the left shows the disease progression throughout the season, with darker colors indicating higher disease pressure. Pockets of disease could form and exponentially increase as one plant sporulates onto its neighbors. Tar spot ratings as high as 40% were observed, but a few plots in white remained tar spot free through the last rating. In addition to tar spot ratings, we measured plant and ear height and recorded flowering time. As shown in the correlation heat map, these traits did not influence tar spot severity, indicating that the fungus was not preferably infecting different maturity groups or plant heights. Here are the results of mapping resistance to tar spot. This graph is called a genome-wide association study, or GWAS. Each dot on the graph is what geneticists call a SNP, or single nucleotide polymorphism. These SNPs are slight changes in the genetics that make up all the differences in the maize lines we tested. At each genetic change, the AUDPC, or disease severity over time, was measured. The computer will check if there's any differences in resistance at each genetic change. Suppose a G in the DNA at the genetic location averages a 20% tar spot severity rating, while a C at the same location averages a 5%. That location would be marked as having an association with tar spot and will appear higher on this graph. Our current data suggests a dozen locations in the maize genome that may confer resistance to tar spot. We are in the process of moving this resistance into more elite field corn and sweet corn lines to fast-track resistance to the grower. We will validate, map, and further explore gene regions linked to the tar spot resistance in the coming years. In addition, we are working to quantify tar spot severity from images, a project that was funded through Bayer's Grants for Ag program. Michigan State is part of the Tar Spot Working Group, a collaboration of teams across the Midwest that are studying multiple aspects of the disease, including monitoring spread, predicting onset, and best management practices. If you have any questions or would like to discuss this project further, our emails are provided here. Finally, thank you for the organizations who funded this project to make this research possible. We couldn't have done this without your support.